CASP special interest discussion on ensembles of conformations, looking at how we might be able to use the CASP infrastructure to evolve toward an understanding of protein dynamics. And yeah, so we organize these meetings every month. A lot of people have been coming. I've already heard this because we do this every month, but this is our vision statement, recognizing that biomolecules do not have static structures, but rather adopt a distribution of states. The CASP SIG aims to evaluate if a critical assessment of these predictions of multiple conformational states is feasible. How do we do it? And considering the new developments, particularly in the area of multiple conformational modeling with AI methods, it seems it's timely to try to explore this challenge. And what we're talking about are different conformational states that a protein might adopt. It could be nucleic acids, but it's not our focus currently. A typical case is adenylate kinase, where there's two different forms that have been crystallized in a canonical two-state conformational ensemble, but we might have three-state, five-state, or many states, like in intrinsically disordered regions of proteins. So we have an organizing committee, and the people are listed here. Actually, this was very much motivated by discussions at the last CASP meeting, with encouragement from John and Christoph and Andre. And so the idea is to actually do a CASP-like experiment, or a CASP experiment, with multiple conformational states. There are actually, I just to point out, I think I didn't put the most latest one up here, but we do have a SIG on AI methods more generally, and also a SIG on RNA and RNA aspects of CASP. So today, Susan Tsutakawa, who's at Lawrence Berkeley National Laboratories and works closely with John Tainer, who's a coordinator of the Sybil's beamline there. She's had a lot of experience working with CASP over the years, and also thinking about multiple conformational states or multiple oligomeric states of proteins using small angle X-ray scattering. So Susan will present, and then we'll have a general discussion about how we move forward with CASP-16. And I should say comments are welcome in the chat. The chat is they're being recorded. The video is being recorded, and it's available on YouTube. And if you want to be on the mailing list, you can mail to that address with that subject, and we'll put you on the mailing list. And there's also a LinkedIn group. So Susan, do you want to then go ahead?
Yes. You have to stop sharing. Oh, there we go. So let me, I have to always move things, rearrange. Here, there we go. And so I'd really like to thank you for this opportunity to give the case for use of small angle X-ray scattering. And, you know, when I was preparing this talk, I really liked how John Molt set up the ensembles. And this really told me, you know, showed me the diversity of ensembles that are possible and that are used as mechanisms by the cell to carry out particular functions. And so I hope that I will touch on many of these types of ensembles and how we've been applying small angle X-ray scattering to understand how these proteins work. And so first, I'd like us to all get on the same page as to what small angle X-ray scattering is, and it is the distance information on proteins. And so I start off with this very simple analogy that if you take, if you average all the stills of this movie of my hand clenching, what you'll get is a very good high resolution image of my palm and thumb, but you'll chop off all my fingers because they're noisy and they're blurry. But as you all know, fingers are very important functionally. And so in the same way, they're functionally important for proteins. So if you then think about the bones in my hand, the bones in my hand are still being retained. And that's what small angle X-ray scattering measures, is the distances between electron pairs. And then they give out a distribution of those distances. So now if you look at a still hand of my hand clenched and my hand extended, you can visualize how that distance information distribution is going to be different. Even though the mass of my hand is the same, the distance distribution will be different between the clenched hand and the extended hand. And in the same way, those distance distributions will be different than my moving hand. And this is the premise of how small angle X-ray scattering works, is that we collect all those distance information and we can differentiate between different conformations. And so to go back to the method, what we do is we take our sample, which is in solution, proteins in solution, or RNA or nucleic acid, and and there's no label, no preparation other than monodispersed protein. We irradiate that and look at the scattering of the X-rays, and in that the information, the intensity is information on these electron pair distances. And so this is very analogous to what you do for crystallography, but instead of an ordered array, we're looking at intramolecular distances, although they're random orientation. So we can represent the scattering of X-rays as a function of intensity relative to scattering angle. And so, and this intensity is on a log scale. And so we can look at it in reciprocal space, but we can also do a Fourier transform and look at it in real space. And this is sort of like looking at density. And this is the distance distribution. So if an electron pair distance is at a particular distance, and then this is its relative proportion among all the distances, and then the D max is the largest distribution. So if we look down here, you can see I've drawn little arrows to represent the distances, and then the turquoise arrow is the D max. And that is what we're looking at. So what do scattering curves look for different shapes and different sizes? I'm showing 3 representative proteins, and you can see that they all have very distinct scattering curves, and they have distinct, and in the P of R plots, you can see a distinct conformation. And then you can see for a PCNA, which is an unusual protein with a cavity in the middle, you can see how it picks up the distances that are within each subunit of PCNA. It's a homotrimer, but it also has some more distances that are across the ring. And then there is a gap in between because of the hole. So this shows that SAXS can provide information about the conformation and structure and solution. Oops. Oh, there we go. But what is really powerful is that we can compare the atomic model to the solution structure. And so these are showing the crystal structures compared to the scattering curves. And so we can do this and show that if we have a really globular stable protein, it matches the scattering curve. And this has really opened up to going from sort of blobs and shapes to actually differentiating between atomic models. And so here I'm going to show a series of examples of induced conformational change followed by small angle X-ray scattering. The first is this is one of the first samples at our beamline, and it's looking at this hormone receptor and a very small molecule. And what you can see is that there are significant differences between when the protein is an apoform versus the ligand induced, and we're able to really observe those differences And we can also differentiate what occurred in the crystal structure, which is I think it was symmetrical, and then one was asymmetric, and we could differentiate what was happening in the so crystal structures with and without this hormone was asymmetric, but in the SAXS showed that there was asymmetric dimer in the apiform, but a symmetric dimer with the hormone bound. And so we were able to observe these changes, and that's because we're not looking at small distances, but we're looking at many distances that are changing in distance. Here's another example, and this I'd like to introduce heat maps to you. And heat maps were developed by Greg Hura as a way of comparing many SAXS curves with each other. It's very difficult to do it pairwise, and so he thought, well, let's just throw everything against everything and color it. So red is similar and white is dissimilar. And so we were able to really follow this monomer dimer switch that occurs, and we're able to look at all these different mutants, or we can look at it with and without ligand and show how the conformation changes or doesn't change conformation depending on our mutation. And in this case, we're following a monomer dimer transition. And although one could see that the dimers are very similar to each other, there are sort of mixed that are in between, that are not just simply switching it from monomer and dimer, in 100% conformations, but there are probably some mixture of monomer dimer that's occurring. And so we're easily able to sort of pick that up. Here's another example where we're looking at a very large, over 100 kilodalton protein, and we're looking at the impacts of small ATP, ADP versus DNA, which is quite large. And what we found here was that that the ATP, ADP was more definitive to the change in conformation than the DNA itself. And so here we're showing another sort of looking at this small molecule induced conformational change. Here goes to this the part in John Moll's plot, which is talking about the impacts of the conditions. And this is a caged protein from developed from the Baker lab. And this is showing a whole range of conditions and trying to really understand how to stabilize an open versus a large conformation. And here we're following many different conditions. And so those are sort of looking at the experimental curves. But what we'd like to be able to do is really represent those these conformations. Either obviously it's nice to have single conformations, but if you have multiple conformations occurring, how do you represent that? And there's two ways to represent that. This way is developed by the Hamburg groups under Dmitry Svergin. And this is what they do is they do 10,000 independent conformations and look at it relative to the small angle X-ray scattering data and group them. And this is probably representative of what occurs in solution, but what is very difficult is to sort of wrap your head around that. How do you understand that? And these are sort of ways that they're using to try to look at 10,000 conformations at one time. Mikhail Hamel in our group, now with Dina Steinman, has really developed this minimal ensemble approach to looking at small angle X-ray scattering data. And that is, what is the minimum number of conformations needed to reproduce the scattering curve? And so this is not to say that this minimum number of conformations are the only conformations that occur, but they are representative of the ensemble that's occurring in solution. And with this, we're able to really see, specific conformations. And this in this case with ubiquitinated PCNA, we were able to provide support that there are actually two dock positions, that it's not just an on-off, but it's, you know, on in position A, on in position B, and off. And we're able to really see that using this minimal ensemble search. And it's not clear to me that we would be able to really pick that up in this, in his 10,000 confirmation thing. So what I'll be talking about as I go further is analysis with the minimal ensemble. So if crystal structures can match the SAXS data, accurate predictions should also match. And we tested this in 2016 and 2018 with the help of Andre and Christoph. And this is showing what we found in 2018. We looked at 11 samples. This is the data in reciprocal space and real space. And we compared that to their crystal structures. If there were disordered tails, we added that in and we allowed them to move to best fit the data. But even with that sort of allowing for movement, we did see significant differences between what we saw in solution and what we could recapitulate in the crystal structures. And so here is showing the 11 different structures that are shown. The stars show structures, crystal structures that match what was occurring in solution. But these all other ones required some kind of movement to occur. And these were not disordered tail movements. These were actually within the globular region of the protein, would need to be adjusted in order to fit the scattering data. And so, and this was also seen by Gaetano's group much earlier by NMR, that he also saw over half of the proteins that he looked at are different between the crystal and solution. We've also looked at CASP 2020 targets, and this was done unofficially because our beamline was shut down during the pandemic. And so we have some samples, and I wanted to go over these a little bit with you. This is target 1052, you know, is a quite large protein, 832 amino acids. And I'm showing the crystal structure relative to the AlphaFold prediction. And it matched very well, with the exception of this petal domain that seemed to be rotated. And the petal domain is where there is enzymatic activity, and so it's very important to understand the petal domain. But also this protein was a trimer, and as far as I know, prediction of the oligomerization state is yet to be proven accurate. But this is showing the trimer, and so what I did was I took this AlphaFold prediction and I overlaid it and created a trimer from the AlphaFold 2 predictions, and then I looked at it compared to the structure. And here I'm showing it both in reciprocal and in real space. And this is the residual, which is the difference between the crystal and prediction. And then here's the real space map. The black is the experimental data, the color is the crystal or the prediction. And what you can see is that not only are there differences between the crystal in solution and prediction in solution, it appears that the prediction better matches the crystal structure than it matches the solution data. And so this is not unexpected, given that the AlphaFold is trained on crystal structures primarily, but it is a point that you know, this is showing the support for that perhaps there is strong bias of the prediction to mimic the crystallographic solution conditions. Oops. Here is another example where the predictions were closer to the crystal structure. And you can see in this case, there was no prediction of the full-length protein, but there was a prediction of half of the protein. And you can see very well how AlphaFold 2 really matches the experimental structure very well. And then when we looked at the solution structure, again, we didn't have the full length AlphaFold 2 prediction, but just looking at the crystal structure versus the solution data, there are significant differences between them. And when we manipulated using Allos mode with small angle X-ray scattering, we were able to obtain a better fit to the solution data, but it required this sort of large sort of curling of the protein, and suggesting that the protein needs to have some population that's in a curled position. And this also brings up this idea that I've been seeing, is that we don't just see conformational changes, or there's this trajectory of conformation. But what I often see is that the majority of the conformation is similar to the crystal structure, implying that a segmental flexibility, meaning that some conformations are stabilized within the trajectory, and those stabilized conformations probably represent functional states. And so we see that in our SAXS studies. And this is just sort of to hammer this point that if AlphaFold, if a group of scientists are asked how do birds fly, and they eliminate all the low resolution data, then they'll get a very nice depiction of a bird sitting, but they really won't understand how the bird is flying. And so these protein structure predictions need to include a more solution-based training as well to really capture functional states of the protein. And then I'd like to make a small diversion, because also in this CASP data set, we observe particular cases of geometric strain. And so here is one of the targets. It's actually ASCC1. We had a 1.1 angstrom crystal. You can see that the AlphaFold 2 really got the topology very nice. But when we did a deep dive into the protein, we observed that this histidine was predicted very incorrectly by AlphaFold 2. And this experimental conformation was reinforced by packing between side chains and an ASP-His pair. And when you take this and you put this histidine rotamer in there, you have to move all these other residues away, and then there's small impacts on the backbone of the protein. Why does this matter? This is one of the most conserved active site residues, and all these residues are active site residues. And this protein, when it's overexpressed in pancreatic cancers, shows a higher risk level for patients. And so you'd want to get this active site correct, say adding an inhibitor, and so you'd want to have this particular active site correct. Why is that? Well, it turns out that AlphaFold 2 was predicting the closest rotamer, and that this particular histidine is a non-canonical rotamer. And therefore, it is in a strained but allowed conformation for histidines. And so, this idea of having unstrained geometry is problematic for the AlphaFold predictions. Here's another example of the Raman-Chandran outlier. This was another CASP target. And what we saw is that there was here's the omit map, and you can see that the experimental model in green is shown here, and then the AlphaFold model predicts this. And why does it predict that? Well, if you look at the allowed rotamers, the allowed Raman-Chandran angles, it is outside of the allowed outliers. But clearly the electron density of the simulated annealing emit map says that this is a true Ramachandran outlier. And this particular part of the protein is involved functionally in a multimerization portion, which has an unusual structure. And so, Jane Richardson is one of the experts in understanding all these canonical side chain rotamers as well as outliers. And she wrote that the few valid outliers are likely to be functionally important and that the protein has a reason to spend energy on them. How frequent are rotamers? She estimated that it was probably about 3 out of 1,000 residues are expected to be valid rotamer outliers. And then Ramachandrans are more rare, but they occur in maybe 5 of 10,000 residues. And so, I think that prediction programs and, in fact, molecular dynamics programs really need to be able to deal with these outliers. And I bring up this quote from I paraphrase it from Bragg Eichmann. He said, AlphaFold 2 predictions are amazingly correct, except for the important bits. And so I think this is becoming a more common occurrence. So the good thing about SAXS is that it's available free at most of beamlines. For us at the Sybil's beamline here, you can email us your samples. It provides strong assessment of stoichiometry, flexibility, and assembly. However, it's only as good as its atomic models. It measures the distribution and distances and compares that distribution. So the atomic models, have hopefully a unique, based on the amino acid sequence, have a unique distribution of distances. And so we can compare that with the small angle X-ray scattering. But here in 2018, what we also observed is that you can take a completely incorrect or mostly incorrect structure, and then you can make it, you can destroy your secondary structure and make it so that fits the SAXS data. And here is showing you the overlay that this is the pre-SAXS and this is the post-SAXS, and for 2 different groups. And you can see that there's a really nice fit to the scattering data. Oh, how do I get this out? I don't know how to, there you go. And if you really zoom in, you can see that the fit is really good, and it's really hard to look. If you look at the residuals, there are apparently biased residuals, so that's indicating that there are differences, but it's really difficult to pick up. So how does the crystal structure match up? The crystal structure also matches up very well with the SAXS data, and you can't differentiate it between the squashed prediction in the crystal structure. Although the residuals are slightly different between the experimental data and the crystal structure. This is in real space. In real space, we do see that there are differences between the atomic models and the experimental data. The experimental data is again in black. And then and here's the similar comparison with the crystal structure. So I think that if we were going to do a comparison with the using SAXS, then perhaps we should do a combination small angle X-ray scattering and something like a sphere grinder type assessment to make sure that the geometry is appropriate. I keep doing this. And then the other caveat to SAXS is that it is an averaging method. We average all the distances in the solution. And so as your that we do see a reduction in the resolution, very small differences is reduced when you have multiple confirmations in the data. So this work is done with help from members of our beamline, led by John Tainer and Greg Hura and Mikhail Hamill. And of course, a lot of this work is dependent on the CASP committee, particularly Christoph and Andre and John Molt. And then some of the samples were provided either by John's group or Andrew Lumbering or Mark Van Rosch. Thank you very much.
Well, thank you, Susan. That was great. I think what we should do is we should have a little bit of discussion and questions around the presentation, particularly around the concept of how we would use SAXS in a multiple conformational CASP experiment, or just a question. And I just wanted to start it off by saying that, a simplistic view is, look, you just have this curve. And with this curve, how can you determine a structure? And I think there's actually truth to that. And there are many, in fact, in principle, many possible structures that could fit the curve. But I think where a lot of the power of SAXS comes in my thinking, is getting relative positions of chunks of the protein right, like relative domain orientations. And so it's actually a very suitable technique as a hybrid between modeling and experiment. The experiment can't give you the whole picture, and the modeling might not necessarily give you the whole picture, but it's a very good marriage between them. And maybe you want to comment, or you or John, on that kind of thinking, or where maybe I'm missing something in saying it that way.
No, that's a great point. But I mean, to me, consider crystal structures. Even there, if you didn't include the stereochemistry, unless you had a better than 2 angstrom structure, you'd have lots of errors in bond lengths and angles and dihedrals and things. So in SAXS, we include a lot of things, including the connectivity and other things, which helps a lot with the histogram of distances. But I think pairwise distances intrinsically give you a lot of the information on the solution ensemble, which is kind of the frontier for structural biology. I mean, consider how much better cryo-EM got when you could put in computational folds and you didn't have to try and trace the chain with the cryo-EM maps. So I think crystallography would also benefit from having these regions that are conformationally flexible, or where there's bound solvent and things taken into account by things like X-ray scattering combined with computational. So I think it's a great opportunity and enabling kind of data to have these distances despite their limitations. Anyway, I mean, I'm very excited by the computational potential, and I think SAXS could contribute to that.
Yeah, so let's take some more discussion and questions. I see David Bell has a question. David, do you want to say that question?
Yeah, thank you, Gaetano. I was a great talk. I really enjoyed it. I was just curious, what is the highest temperature that you can do while taking SAXS measurements?
So, in theory, we can take fairly high temperatures. The rate limiting step is obviously you want to prevent the vaporization of your sample. But previously, the PCR machines, we had hooked up a PCR machine to our sample cell, and so we could vary the temperature very quickly. So we're doing that, but it's very on a case-by-case basis, and it's typically, I think we're going up to 50 degrees Celsius. Not really high temperature, but the potential is there to do that. It's just trying to get into the high throughput and routine measurements
I
has
think,
led us to adopt a different design.
I mean, David might have a great point. What was your view of the temperatures? Were you thinking of comparing things at, say, 5 degrees and 20 degrees and 50 degrees or something like that? Or what was your thought behind that question?
I was curious about like the melting point of proteins and maybe like going up to like 75 degrees Celsius or above if that was achievable.
I mean, with a Peltier device, you could do those things. You have to, because we're collecting data pretty fast now, I think the problems of evaporation might be surmountable. What do you think, Susan?
They are surmountable. The bigger problem is denaturation of the protein on the surface of the window will be an issue. Kind of like bugs on your window screen. And we do have, with just radiation, we see that. So presumably at 75 degrees Celsius, if you've got that, then we're probably looking, that might be an issue of looking, for unfolded, molten globular type things.
I see.
I'd like to ask a question now if I can, which is, and people who want to ask, just turn on their video so I see that you want to ask a question. It has to do with the time scale. So if I have two, let's say, very significant conformations, and they interconvert slowly, then I guess my SAXS curve is going to be the population weighted average of the scattering from each of them, I think. But on the other hand, there is some time scale when if they interconvert faster than that, then the scattering that you measure is the scattering that's the average distance. And I think that time scale is very fast, but do you know what that time scale is? Where
No, what we do,
slow exchange and fast exchange in SAXS?
yeah, I think what we'll see is just the not an average of the two. It'll be because you can imagine even the transition is occurring, in the millions of particles in your sample cell. So you will see,
It's a sum, not an average.
yeah, it's a sum of the different conformations occurring.
Nothing that's right. In SAXS, you'll see the whole population ensemble. It's just you'll see the sum of those distances.
There must be some time frame, and you may not see it, where when you interconvert faster than that, then I think it has to do with the lifetime of the excited state. Like in absorbance spectroscopy, the time scale is nanoseconds or femtoseconds. And in NMR spectroscopy, it's microseconds. Do you know what it is in SAXS? So I understand what you're saying. You will see the sum. You'll see the population, the sum of the two spectra. But at what point would it be average? Or nobody ever thinks about that?
Well, I guess you're envisioning that there's a conversion that's relatively slow rather compared to the occupancy of the states. Whereas I imagine that those switching between states is concerted and cooperative and happens pretty fast. But we're measuring data at the we can measure what, 30 millisecond time
Yeah,
frame.
we're at the millisecond time frame.
So most of the allostery we do is slower than that, I guess. But maybe, we can say, look at ATP hydrolysis, or we could use photo caging or things to have things in one state. And then I think people have used SAXS, Susan would know more about this, to look at folding events, which would be a case where you want to see the kind of question that you're asking. Do you see averages in a folding thing, or do you actually?
Yeah. I mean, I think it's just a sum of the conformations. But I to go slow, we have SEC SAXS, so size exclusion coupled SAXS. So, we elute right into the SAXS sample cell. And in one case where it appeared that the exchange was at the minute timescale, we could separate out. So it looks like one peak in the OD 280, but it was two populations, and we could see that those different radius of gyration separate out.
Can I weigh in here a little bit to help explain Guy's question? Because I think you guys are not quite following his question.
Thanks, Terry. Go ahead. Yeah.
We had the same concern when we started doing SAXS, and it's something that NMR spectroscopists always have at the front of their head, because in NMR, relatively biologically significant conformational changes take place at a time scale that has a big impact on what we see in the spectrum, okay? So when two different conformations interconvert on a microsecond to millisecond time scale, we see big differences in the spectrum. Now, when it comes to scattering, the question Guy was getting at is, what is the time scale of the initial scattering event that occurs that then gives rise to the scattering that you actually observe? And I think the answer to that is it's way short. It's down in the femtosecond range.
That's what I
right?
think. Yeah.
That was the conclusion we came to, Guy, very early on, is that it's basically a snapshot. It's the closest you can get to a snapshot of the populations.
Yeah.
So
For biology, it's instantaneous, I guess.
Yeah.
yeah.
Yeah, that's the question I was getting at. If there was an interconversion faster than the time scale of the scattering event, then you would actually measure an average distance.
Right. But that's impossible, Guy.
But
Just can't happen.
Yeah. This time scale is like absorption spectroscopy kind of time scale that way.
Right.
It's I don't know the number, I was so asking it, but I guess it's picoseconds, femtoseconds, so that when modelers make structures, we can assume every structure in that ensemble that's made would be contributing to the experimental SAXS
In
observation.
proportion to its population.
And that leads to Keith's question. Keith, do you want to say your?
Actually, Arnie's been very patient, and then
okay, I didn't
also
see that.
I have to
Go ahead, Arnie.
apologize because I have to go now. So John will take over. Thank you, everyone.
Yeah, there was a conflict. Arnie, do you want to ask your question?
Yeah, I mean, I was just thinking from the CASP perspective, like, I think there are two aspects that we can use SAXS for. One is basically to distinguish cases where we don't know the biological ensembles, like we can do a big complex and we don't know the orientation. And the other one is basically for things that are more or less dynamic, more or less somehow disordered, but it might not complete. And then we can provide a lot of models, we can see whatever fits. I would imagine the first case is rare, that is really in CASP, but it's that. So it's certainly interesting from many perspectives, like, but I don't think that really would fit into the CASP thing, because the cases where the cryo structure is not good enough and not biologically correct, and we don't know a much higher resolution data, going to be few. But the second case, I think, is the interesting one, where we basically have something which is very flexible, dynamic, not completely disordered, but where a set of models can provide insights from the SAXS data. So is that what you imagine we could use it for? Or
Yes, I think so. And I think this is the crux of it, because those dynamics are often the functional bits, right? That this is the reason, as a crystallographer, I struggle so much to connect structure to function. It's not so easy with crystallography, but when you include the dynamics, then the structure really does tell you what's a possible mechanism and activity and what's not. And so that dynamics is really the key, right, to linking the structure to the function. Don't you agree?
yeah, I agree. I agree. But the question is, how would we do that in the CASP perspective? So would people submit 1,000 models, and you would take the average of them, or X number of models with some weights, and then you see whatever fits the data best? Is that the way to do it?
I mean, I think you guys are in a position to help guide this, but the way we've been doing it is we look at a whole sort of heat map of different conformational states, and then we cluster them. So we say these states, these mutations or these ligands bound are more like state A, and these other ones are more like state B, and so forth. So we've used this, for example, to do screening for inhibitors, and we can imagine locking something in an open or closed state or somewhere in between, or locking it so it's flexible and can't become ordered. So grouping things, clustering, ways to cluster things based on the SAXS distances is one way we've been doing it. But I guess what we'd like to know is, if suppose you have some cryo-EM structures, but you think there's some intermediate state you're not seeing. Well, the SAXS would tell you what that state might look like and where it would be relative to the distance states that you have. So you might find that there's something that's very different than any of your structures that you're missing that really happens in solution. And so I think ways to think about that and ways to consider how to do that rigorously are, I think your question really hits the nail on the head, that this is where we need to do better, I guess. But
from a predictor's perspective, I think the easiest way what people do is basically submit X number of cluster centers with some weights.
Right. So
and then the SAXS people have to evaluate which prediction is the best.
Right. So you might want to say there are three major conformational states, and these models are missing one of the three, or you've got all three major ones, and this is great. Yeah, because I think that can really help link structure to function, and with the design of inhibitors and lots of things. Like if you understand which things have to move and you can lock them down, you've got an allosteric inhibitor, and so forth, or you've got a way to evaluate cancer mutations or whatever. So I think these kinds of questions you're asking are really valuable.
I think, Keith, you had a question. Why don't you go ahead? I think it's very similar to what Arnie was just saying, but go ahead and make your point, Keith.
Yeah, I mean, the point is really along the lines of Arnie's second goal in assessing conformational dynamics in CASP using SAXS data. I mean, I think Susan had a slide earlier about two different ways of building ensembles. There was the 10,000 structure way, and then there was the minimal ensemble way that Susan led, my understanding from the presentation. If there's a way to sort of give a score or a number for a given ensemble and a SAXS distance distribution, is there a score that the ensemble agrees with that distance curve? And then you could have predictors submit multiple models for a target that has a SAXS data behind it that they don't see.
Yeah, that is definitely the idea. We will assess predictions against data.
Yeah.
And we're going to need John and Susan's help to do that. And I think you also brought up the question, how do you structure it? Well, the modelers will predict a set of structures and their populations. That the populations have to be part of it. And then going to Arnie's point, maybe we don't want the 10,000 models. Maybe we want a limited number where they've clustered their results and provided up to, I'm not sure the right answer there, actually. So I thought that's one thing you were going to get at. How many?
Yeah, my idea was that you penalize the predictor for submitting more and more models, because you probably can arbitrarily get close to the distance curve if you just send in more and more models. So some way to sort of downweight a predictor submission relative to the number of models that they submit is what
I do think we have to think about that. I'm not sure what the right answer is, though. Terry, you're shaking your head. But you're on mute. You're on mute.
I strongly agree with you, Guy, that one concern is this population thing. So the SAXS data will definitely contain the relative populations of all the conformations, okay? And yes, they need to be clustered in some kind of sensible way. I think that's going to be a huge challenge for the predictors, is to get the populations right. And so one question to ask is whether CASP will score results based on just the fact that, yes, they saw approximately the member conformation that the SAXS data seems to support, but they didn't necessarily get the population right. I mean, have you thought about how the population prediction is going to play into trying to predict the data?
I wonder if the population should be a separate issue, because as a biologist, if I know that there's only two conformations, say there's a closed conformation and then there's a flexible conformation, there's not really a stable open conformation. That's very useful information. And I may not care how much of it is, because that may be situation dependent. It may depend on, you know, partner proteins or RNA, DNA or ligands or all sorts of things, right? So in the cell, or in, you know, these populations may not be the population of the individual complexes or things. So it'd be more valuable to me just to know there are 3 states or 2 states, and what their populations are is probably context dependent.
Well,
Right?
it's absolutely right. But a specific question is, how do we assess a prediction? And we want to assess, we could assess a prediction against the experimental models as ground truth. But I think we've agreed in these discussions, the experimental models have to be made from the data with some multiple modeling technique. So is that really a ground truth? So I think that we've decided the ground truth should be assessing the predictions against the data. And what we're getting at here is, to fit the data, you also have to know the populations. So one way to do it is predictors give us their N, I don't know what the right number for N is, their N clusters, their N representatives, and their populations. We might, depending on who's involved in the assessment and the resources we have, in assessment, allow the populations to float somehow, to say, given their 3 conformations they predicted, if I use their populations, it doesn't fit so well. But if I float it so 2 of them are there and the other isn't, is very low, it fits. Now, I think that would be the right way to do it, but it'll take some resources to do that. Did someone want to comment on what I said, and then we'll move to the next question?
Guy, I was trying to think in terms of the targets we had for multiple conformations last time, what might have worked. And the only one I can think of is the ABC transporter. So this we had in something like 5 different states, which turned out were 3 different conformations with different things bound. But in this case, you could, in principle, I guess, have done the SAXS measurement on all 5 of these assemblies. I mean, it's just a small molecule and a large molecule, and asked whether or not the predictors picked out the right conformations for each binding. And that's there you don't have this problem of weights, right? You're changing the conditions to sample different conformations.
Those are good kind of data to do too. I agree. If we
That's
could,
a more biological kind of question. So I think that's quite valuable.
well, maybe this kind of leads into, if I can, I just want to just flip a couple more slides up, which I have here, about how we go forward with this experiment, and hope maybe we'll have also time to come back to some of the other questions. Let me share the screen, because I do want to end on time. And see here. So we're evolving toward trying to do this experiment in cat, to really do it. And it's very challenging, and it's only CASP 17 where we really pull it together. But the idea is we have some ground truth data, and then like, let's say we have SAXS data for a system, and we release the sequence to the community, and they have to predict a distribution of conformations that we would then assess against that data. Now, it might be the experiment is we have three different conditions where we blocked out different conformations. It would make it easier. So this is kind of a plan, and I should say we've been reviewing these ideas, both the prediction methods, AlphaFold sample and AlphaFold alt and AlphaFold cluster and other methods. And we've been predicting the experimental things, like we've been exploring the experimental things like cryo-EM and NMR relaxation data and SAXS now and high resolution crystallography. And those are discussed in these videos that are available. And now we're looking at, what kind of, where we can actually get data to do the experiment. And Terry Ois is on the line, and Bruce Donald, his collaborator. They've been working with a system which is a 2-domain system connected by a linker. And they've been doing a very cool kind of NMR experiment, a residual dipolar coupling where you orient one domain and see how it affects the dipolar couplings in the other domain on their system. And it's also possible perhaps to get SAXS data. And in our meeting next March 6th, Terry and Donald are going to describe their data. And I hope what will come out of that is a data set for CASP-16. Maybe it's a SAXS data set, or maybe it's an RDC data set, or both. This is actually a system we work with in my lab, and we've been doing similar things. We actually haven't got that RDC experiment data working well yet, but maybe we could use this system also for SAXS. And so now it brings to this point, like, how do we get more data sets? And how do we, I think one thing is I could write a letter to particular people, or if you could suggest people, so this is really what I wanted to come to at kind of the end of this discussion now, is to get some suggestions how we could drum up more data sets. So maybe John or Andre or John Tainer or John Molt have some suggestions about what should be, or other people, next steps in actually moving this forward.
Well, I don't know, Guy. I think the compiling a list of people to write to, I think Andre's the expert on this, but those sort of personal approaches are much more effective than putting out some sort of general on a bulletin board thing. And so if you could utilize the people in this group to suggest what you were saying, right, who to write to, that's likely to be productive.
Yeah, what I will be doing, I'll be polling my network of experimentalists from different experimental techniques, and they have contacts with more than 200 people all over the world, so that's what I'm going to do also for CASP-16, but that's not targeted for specific, kind of targets that we need here. So as John said, so if we have specific contacts to people who do these things, so that would help immensely, because from my experience, the personal contacts work the best. Just casting wide nets, maybe one fish for us, what we want to have.
Can you help me think about this? I'm just imagining, like, the Kramer lab or the Eva Nogales lab. Some of the cryo-EM labs probably have things where they have a structure and then they have pieces that are undefined or flexible. Would those kinds of data be helpful, or what could you get from EM labs that would be useful? What are you guys envisioning?
Well, we had Wachu present in the same way, and Wachu, I wrote to him a while ago, and he said he would come up with some targets.
Oh,
so
great.
Follow up. But who are some other names you're saying? Kramer is
Patrick Kramer, spelled with a C, and Eva Nogales, Yuan He. I mean, I could send you, say, there are probably fewer than 10 labs that are producing a lot of structures of sort of dynamic complexes involved in transcription or replication or and so forth that might be useful, depending on what you want from EM, I guess.
Yeah. So I will prepare specifications, and Wim Ronkin asked me to do this already, of what we're looking for, and I'll share that with Andre and get some feedback from some people on the committee and even on this call. And then I'll take that same list and try to go to specific labs. And I mean, I guess I certainly could, I know people who I might approach, but in the last few minutes, maybe I can get a few more suggested names from this group of who I might approach about this.
I put some name in the chat.
Thanks, Arne.
I do believe that probably the most valuable information would be cryo-EM, because that's certainly many more people doing cryo-EM now. But Kirk has had his hand up for a long time. Maybe we should give the word to him.
Yeah, go ahead. I can't see whose hands are up the way I'm writing now.
Oh yeah, sorry about that. Yeah, I guess for like, 2 points now, given you brought up Wachu, if you really want to ask him, maybe like ask him about some of the hinge glycan data that was recently produced and he put out like last year. That might be like a useful, relevant data set and something worth that, more global interest could be paid attention to. But given I was sort of late for some of the SAXS conversation, I just wanted to ask, like, did anyone want data that was like SAXS compared to wax, or for combined SAXS and SANS for small angle neutron scattering? Because like sometimes there are some labs that will generate both combined SAXS and SANS. And so I was just sort of and that's like for a lot of proteins and for like protein RNA combinations, and being able to use SANS in order to do better resolution of the solvent environment. So I was just sort of curious as to whether or not, doing imaging and pulling out the data with like combined protein conformation structure and the solvent environment would be useful. Because like some of that's already been done. So I was just sort of curious as to whether or not just asking them to like generate some more would be productive.
Yeah, I think the thing is, I can try to solicit such experiments, such data. The people who generate the data are probably going to have to be involved at some level in the assessment. So, because I don't think I'm in a position to assess SANS data. So, I mean, so I think it's worth bringing up. And if Susan is going to be involved and help with assessment and she's okay with SANS data, we could do that.
Okay.
Do you have a particular name of someone I might want to put here?
Oh, just to do review of SANS data, I'd have to look up whom I've talked to at Oak Ridge about some of this stuff.
Who I would approach to provide data for a CASP-16 experiment that would be done this summer.
Okay, yeah, let me see if I can review some names, and I should be able to like email them to you, I think.
I mean, obviously the Oak Ridge guys are, leaders in this, and one area that they work on are things like the COVID viral proteins, and thinking about the polymerase, there's cryo-EM structures with the RNA and all the subunits, and there's SAXS data on all those independent things and how the different subcomplexes bind or don't bind the RNA and what they look like. And then I think Oak Ridge is getting SANS or has SANS data on those things. I don't know if that would be a system that would be worth doing computational populations on it. It's obviously very relevant. And I think there are many mysteries involving these RNA polymerase, these viral RNA polymerases, as to how they work and regulate. You know, they're doing transcription and replication essentially. So there's lots of molecular gymnastics there. I don't know if that's off track for you guys, but
Well, that hinge glycan that I was pointing out for Wachu's work, that was from SARS-CoV-2 spike protein hinge glycan specifically for like
right.
multiple
Yeah, that's brilliant.
strains. Yeah.
Well, I think we should stop now because I told people we would try to keep this thing cut to time as best as we could. And if you have additional suggestions, if you could, there was actually, this video will be available soon online. And there was an email I put in there that you could email related to this group. And, or people can email me directly, people who know me. I tried to put together a solicitation to share with Andre that he could share with his network. That's probably where we'll get the most samples. But I can also approach individual labs this way. I think we have to move ahead to start to get targets for the summer. Actually, so next week, not next week, next month on March 6th, Terry and Bruce will talk about their experiment and their data. And I'm hoping we could at least get SAXS data on their system, if that can work. And maybe also the kind of experiments Terry's been doing with RDCs. So is that a good point to stop now? Does anybody else want to say something before we end?
Thank
Okay.
you guys for the input on the SAXS.
Thank you, John. Yes, thank you, John. And Susan, who's off for the presentation. Thank you very much. Thanks. Okay.